Log on, tune in, find out. Another good idea from Cambridge.
The first of which is given by Sebastian Ueckert from Uppsala University in Sweden, and he's going to be talking about the optimal design of clinical trials in Alzheimer's disease.
Thank you. So my name is Sebastian Ueckert. I'm a PhD student from the Pharmacometrics Research Group at Uppsala University, and I will give this talk in place of Andrew Hooker, who unfortunately can't be here today. I will talk about a clinical application of optimal design, and I will explain the benefits of optimal design in this setting and also the challenges that we faced when we implemented this method. But first, I would like you to think for a second about important persons in your life. So that, for example, a partner or parents, kids, etc. Now imagine that all of a sudden you can't remember those people anymore. You have the feeling that they're important in your life, but you can't recall their name or can't remember which role they play. Or imagine the opposite role, that one of them can't remember your face. This is a situation that many million Alzheimer patients around the world are facing every day. So Alzheimer is a progressive brain disorder, and it destroys brain cells, which leads ultimately to a loss of brain function. There are several hypotheses what actually causes Alzheimer. One claims that the reduced synthesis of acetylcholine leads to Alzheimer. Another one tries to establish a link between Creutzfeldt-Jakob disease and Alzheimer by claiming that a misfolding of a protein leads to Alzheimer. And the third hypothesis is that a process which is important during embryonal development is falsely activated again and leads to this remodeling of the brain. So you see that this is very uncertain. What is certain is that Alzheimer is the number one disease, the number one cause for dementia in the US. It's the sixth most common reason for death in the US, and while other major diseases like HIV or heart failure have decreased, or the number of deaths have decreased since 2000, the number of deaths for Alzheimer has increased. 1 in 8 elderly Americans have Alzheimer, and every 69 seconds there's a new one developing Alzheimer. Due to the aging society, the number or the costs, the annual costs for Alzheimer is projected to increase to 1.1 trillion in 2050. So there clearly is a need to act for this disease, on this disease. The way to diagnose Alzheimer, there are objective ways. You can diagnose it via imaging techniques like PET scans or via biomarkers, but the most common and most accepted technique are still psychometric assessments. And among them, the ADAS-Cog scale plays an important role since it's accepted by the FDA as a clinical endpoint. It involves 11 tasks which measure different functions or the performance in different tasks, and it's between 0 and 70, where a score of 18 or more is interpreted as cognitive impairment. Very important as well is the mini mental state examination, which is a brief test, and it's usually used during screening. So you saw all those numbers about Alzheimer, that there is a need to act. But if you look at the drug market at the moment, it looks very, very sparse. There are 3 acetylcholine esterase inhibitors and memantine that are on the market. And it's not like that the drug companies don't try to develop drugs for this market. In fact, many drug companies had promising candidates that failed in phase 3. So in the phase where actually most of the budget is spent. So you really don't want your trials to fail. And the reasons for that, of course, they are very diverse and probably a long list. But I found in this article 3 reasons that they claim play an important role why those clinical trials fail so late. One is that animal models inaccurately predict what's actually happening in humans. A second one is that the wrong target population is maybe selected for those clinical trials. And then the third one, that a poor study methodology is used. And that more or less, or fighting that is more or less the reason for this workshop. So I'm sure I don't have to convince you that a model-based trial design can improve the outcome of those trials. The benefits of model-based trial design are that we can do an educated planning of a clinical trial. We can combine information from several sources to create a model. We can evaluate possible scenarios, and we can optimize the trial design. In this project, we combined several information sources to actually create a model that we use then for the optimization of the trial. The first source that we used was a meta-analysis. And this meta-analysis is published in Alzheimer's and Dementia. And what we took, the components for our model that we took from this disease are the natural history and the placebo response. So how does it look in terms of formulas? Since over the time of a clinical trial, the progression is very, very slow and the changes are very small, we can easily approximate that with a linear function. So we have a baseline and the natural history progression rate that describes that. And then that does not look very spectacular, it's just a line, yes. The second component that we took from this meta-analysis were a placebo response at the corresponding parameters. So a placebo response can be modeled with this kind of Bateman function consisting of a placebo amplitude and a placebo onset and offset half-life or half-time. And then it adds up just additively.
Do you call it meta-analysis?
Yes.
I don't think it's a meta-analysis.
The article was clearly a meta-analysis. It's even in the title.
It depends upon definition. So
Yeah, but
They are trying to build a model properly.
Yes,
So
but
what they did in this article was they did a literature research, and then they took parameter values from those different studies and calculated summary statistics over them. So if you look at how this placebo response looks, you see that after the beginning of the trial, we have some kind of effect, but as we wait longer, this effect washes out and we are at the same progression as the natural history. The second information source that we used was a population model, also published in Alzheimer and Dementia. And the components that we took from here were the covariate relationships, the between subject variability, and the dropout pattern. So the covariate relationships that were found in this publication were affecting the baseline and the natural history. For the baseline, it is that a higher or a worse mental state at screening also corresponds to a higher baseline value at the ADS-COG scale, which makes sense. The covariates that influenced the natural history progression rate are ApoE4 or apolipoprotein E4, which is the most important genetic risk factor for Alzheimer. Furthermore, we have that females have a higher progression rate than males. People with a higher age progress faster than people with younger age. And we have this relationship with the baseline here, so that people that have a high baseline also progress faster during the trial. And then on top of those covariates that explain some of the variability that we encounter during the trial, we also have those two random effects on those parameters. So if we include them into our model, we are actually able to describe a whole population of patients. the third component from the population model was a dropout pattern. And this was modeled in a very simple way. So it was modeled separately as an exponential distribution, and it didn't depend on the disease status, it only depended on this mini-mental state covariate, which will help us later when we calculate the Fisher information matrix. So looking at the dropout model, we see that after 24 months, which is a common duration of this kind of studies, we see that approximately 30% of the patients dropped out. The third and last information source that we also used to develop this model is where we took covariate distributions from clinical trial databases. And these clinical trial databases contain more than 3,000 patients, and they contain information what are the distributions you usually encounter when you do a clinical trial in terms of covariates. So then, of course, we also need a drug effect if we want to plan those clinical trials. And we hypothesized one drug effect which changes the progression rate by 10%, which has a delayed on and offset, so it takes some time for the effect to begin and to stop. And the time to half maximum effect was 3 months. So that's really hypothetical, and it can be easily changed if there are different expectations. So a way to model this is that to make this progression rate or this progression constant dependent on time, and then to write it as a differential equation using this kind of effect concentration here. So here in this graph, I visualized it by plotting 30% drug effect, otherwise you wouldn't see much difference. And it also stops after 12 months. So you see that it takes some time it has a bit this S shape. That's how the drug effect would look graphically. So that's the final model, or all components together constitute the final model that we use for the project. So what we could do with this kind of model is clinical trial simulations. The benefits, it's relatively easy. It can be unflexible. We can test a lot of different scenarios. But the limitations is that you can only do a heuristic exploration of the design space. So you have no information. What is this design that I tested? Is it optimal, or how far am I away from the optimality, and so on. And it is computationally expensive. So you can only do it for a certain number of points. Therefore, we resort to optimal design. So what we want to do, we calculate the information matrix, and then we can use different scalar function to find the optimal design over the specified design space. But of course, we have several aspects in our model that we have to handle first. So calculating the Fisher information matrix is not that straightforward in this scenario. So the first thing we have to handle is the covariates. So what we want to do is we want to calculate the Fisher information matrix over the covariate distribution that we have from those clinical trial databases. And there are two ways to do that, or at least two ways to do that. We could solve this integral here with Monte Carlo integration, which has the advantage that we can directly sample from the covariate distribution, so have the true distribution. But it has a disadvantage that we have stochastic noise, which makes the optimization afterwards a bit harder. Therefore, we selected the second method, numerical quadrature, which has the advantage of having no stochastic noise. But we need a probability distribution function for the covariates. And it's not feasible if we have too many continuous covariates, because the dimensionality grows too quickly. So to find the probability distribution function, we simply fitted normal distributions, which in our opinion match those uncounted distributions quite well. And then we can do a numerical quadrature. And in this case, we do a two-dimensional Simpson quadrature for the covariates mini-mental state and H. So we discretize this continuous two-dimensional integral and then replace it by this sum. To handle the dropout pattern, we have to calculate again the expectation of the Fisher information matrix over this exponential distribution of dropout times. And here we are more or less lucky that the parameters for the dropout function are not dependent or are not contained in the Fisher information matrix. So therefore we can simply replace this integral by a sum, and what we basically do is we calculate how many individuals or what percentage of individuals has 6 samples, 4 samples, 3 samples, or 2 samples, and we add them up. So we scale down the information according to the dropout. So after we solve those theoretical issues, we can implement that, and we implemented that in our optimal design software Puppet, which is freely available, and implemented in .NET. So there's a graphical user interface, which makes it easy to use, and the calculations are handled in MATLAB. It has a long list of features. Some of them are model library. We have a lot of different FIM approximations. It supports ED optimal design, and new since this year is we can also do parallel computations with either OpenMPI or the MATLAB parallel computation toolkit. So Puppet also has a high flexibility, so you can write penalty functions which handle these complicated cases of integrating over the covariates and the dropout function. And then the way the communication works in the graphical user interface, or the graphical user interface generates the configuration and parameter definitions according to the user specifications. And the calculation engine takes those values and calls the penalty function and the model function to calculate the Fisher information matrix. Before we actually optimize, we should of course check if the theoretical or the assumptions that we did doing to handle the theoretical issues are justified, or if they lead to a correct prediction of the empirical standard errors. So what I did here was simulated 1,000 times data sets, the clinical trial simulation, and then I re-estimated those data sets, obtained empirical standard error, and then we can compare that with the predicted standard error here in red from Puppet. And you see that the difference for those covariate parameters is minor. So then we can turn to optimization, but then there are other challenges we have to solve, or we are facing. So one of them is that if you just do unconstrained optimization, you often face a situation that you end up with designs that are not clinically possible. So like replicates. Here in this graph, we show that the optimal design could be sample 3 times in the beginning and 3 times in the end. Of course, that's not possible with the same individual. But also sampling at 0, 0.2, 0.1, 0.2 is also something which wouldn't be possible. And the way to handle that is to use inequality constraints, to say, okay, we allow only samples every month, or whatever the requirements are from the clinic. And then in order to solve that, we use a trust region reflective optimizer that can handle those inequality constraints as it is implemented in the optimization toolbox in MATLAB. The second challenge is connected to the delayed onset drag effect. Since we implemented it as a differential equation, the calculation time is quite high. So calculation of one Fisher information matrix is 15 minutes. And then the optimizer that we are using needs a derivative of this scalar function. So we have to calculate, even for one point, we have to calculate the Fisher information matrix several times. So that 15 minutes is really too long. But luckily, we can solve this differential equation analytically, and then the calculation time goes down to 1 minute. And then combining that with parallel computation, we can actually optimize. But optimizing it, this sounds so straightforward. It sounds like, okay, we have the Fisher information and then we determine the optimal design. But as you can see from this graph, that's actually not very clear what optimal design means in a clinical sense. We have a lot of different people that all have their different interests. So we have sponsors that want to have a maximal success. We have scientists that maybe want to maximize the knowledge. We have participants that want to minimize interventions. And we have regulators that want to minimize the risk during a clinical trial. And all of them have different requirements on the clinical trial and on the design. So let's see how we can address that. Minimal interventions. So what we could optimize over is the number of samples in the design, with the constraint that we have at least as much information measured in terms of the determinant of the Fisher information matrix as the initial or the standard design, default design. And then this default design looks like that. So we have two groups, treatment and placebo. The duration is 24 months. And we have sampling at 0, 3, 6, 12, 18, and 24 months. So that's the default design that we use as a comparison. And if we optimize, we can obviously optimize on the sampling times, but we can also optimize on the covariate distributions. Since we have that in the model, we can say, okay, what is the best inclusion criteria for this clinical trial? And then what we end up with is that it's better to have two groups, and in each group a placebo and a treatment arm, and then have different sampling times in those two groups, leading to, in total, 33% less samples, which is obviously good for the patients, but also for the sponsor, since those investigations are quite expensive. The next actor, the scientist, maximizing knowledge. That's maybe more what we are used to, because we can interpret maybe knowledge as information. So we want as much information as possible. So we maximize the determinant of the Fisher information matrix. And then what we end up with is this kind of design. So it's actually optimal to stop the treatment a bit earlier and then to have a washout, but anyway continue observing. So we have samples here, 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 and then two after the treatment stopped. And then you can see, if we look at the relative standard errors, here in gray are the ones that for the default design, and in red the ones for the optimized design. We see that we have a reduction, and on average that's minus 22%. So here I plotted the change from this default design. So the third actor, the sponsor, who might be interested in success, we can interpret that as the probability of correctly identifying a drug effect, or statistical power. And then we developed the extension of the classical Wald statistic, which was presented at the PAGE meeting this year. Which so the classical Wald statistic in matrix notation is this formula, and then we extended it for a nonlinear, or we used a nonlinear formulation of that to optimize directly for statistical power. So we can then plot the power, so the probability to correctly identify a drug effect versus the number of individuals for the standard design. And we see that we need approximately 1,200 individuals for 80% power. And then we can compare that with the d-optimal design, which is the usual way we optimize. And we see in that case that the d-optimal design didn't improve this a lot. So there's almost no change. So if we use this direct power optimization, we end up with this design. So now it's not optimal anymore to stop the treatment earlier compared to the d-optimal design. and the samples move more to the beginning and to the end. And in terms of number of individuals, that's 449 less to reach 80% power, or 37%. The last actor, maybe the regulator that is interested in minimizing risks, he maybe wants to do a data analysis that is with minimal assumptions. So they might don't want to do model-based data analysis. So just as something to think about, doing the least square means analysis in that case would require 1,443 individuals more to reach 80% power compared to the model-based analysis. And if we compare that to the optimized design, it's even 1,892 individuals more, or 3.6 times more individuals. So in summary, I hope I showed you that an application of Optimum design in a complex clinical scenario. You saw how we pooled information from different sources. We handled covariates and dropout, and we used a constrained optimization which was not, or we increased the speed. Also, we illustrated how flexible optimal design can be, so how we can use different criteria to satisfy different actors in a clinical trial. And with this acknowledgement slide, I would like to close my presentation.
We have time for questions.
Wouldn't that be natural to combine some of these criteria?
Yes, that's something we work on.
So you will present it next year.
Yes, if I'm invited.
I soon have some kind of comments, but which needed some kind of response from us people. So actually I like your criteria, and not only me, Walt's criteria, which is related to hypothesis testing. But actually Walt himself, and somewhere in 1943, derived that actually this criteria, he derived the necessity to introduce de-optimality because of de-optimality is equivalent to some extent to his criteria for hypothesis testing. It's how de-optimality actually appears.
Yeah.
So there is no, I am not amazed that these two criteria actually behave very much similar because of this fact. So.
Well, actually, here you can see that in this scenario, it's not the case, that you have a big difference for the de-optimized or de-optimal design versus the
How optimal design, but you didn't formulate alternative here. So if you formulate it correctly, because for calculating power, you need to introduce some alternative.
Yeah, the alternative is having no drug effect. So
It's still not very clear alternative. But okay, in any case, look at this paper by 1943 when actually Walt connect hypothesis testing with deep optimality.
You use D-optimality or DS-optimality? Optimality for that parameter?
It doesn't, if you care about few parameters, then it's DS optimality.
Yes, that's so.
If you care about all parameters, then it will be D optimality. So it's just how D criterion was introduced.
of course, there's a very close relationship between D and this power optimality because they both contain the Fisher information matrix. I mean,
Okay, so just look at this paper.
Yeah, well, maybe I can anyway
I
respond.
think maybe the difference is that the power that he's showing would be equivalent to some version of DS optimality instead of D optimality.
You see, and it's also when actually Walt considered this, it's considered that not all parameters equal, so there are eigenvalues for all parameters. So he introduced that you have to minimize actually sum of eigenvalues or product of, and then you come to some extent to D optimality, which is bound from the law. So this is the first comment.
Just a second. So the reason that here these two criteria are not behaving the same is that D optimality focuses on those placebo parameters, which are don't matter for those for the power. So in order to decide if I have a drug effect or not, I don't need to know I don't have to precise information about placebo. So that's the reason why they are so different.
So
And then
you are right. I just try to emphasize. Look at this paper. Period. So just
Yeah, but I would.
because it's explained connection between hypothesis testing
Yeah,
and
obviously. I mean,
the optimality.
we had to read papers in order to develop this criterion. And in fact, the advantage of this criterion is that I don't have to decide which parameters are important for me. So if I have D as optimal design, I have to say, okay, it's the drug effect parameter that is the most important. And as we showed in a different study in a page, sometimes it's not enough to just know the drug effect parameter. So this criterion has the big advantage. I just specify this criterion, and then it's automatically taken care of which parameters to focus on.
Okay. Thank you. Then I have another comment, and I think it's somewhere in the vicinity, if I'm allowed. It's the vicinity of slide where you did averaging of information matrix.
Yes. For the dropout or for the?
Sorry?
For,
It was somewhere close to what the first thought of your presentation.
so it's
Oh.
average here and
It's okay. Let's start with this average. Okay, so let's assume that your design Xi has one point, and you have 10 parameters. Obviously, you cannot estimate,
yeah.
okay? Because it will be single. Now, if you do averaging, now your singular matrix after averaging, which has rank 1, your average matrix will have rank n, equal to the number of unknown parameters if your distribution P of A is not single.
In this case, the rank of the information matrix may be more than one because you have several
Okay.
samples per patient.
Okay, so if it's several observations, if you observe several responses, fine. Let's assume you have one response for the sake of simplicity. So that is why your optimal design, in this sense, optimal design, may have, say, 1 point, theoretical, or 2 points, when you have to estimate 10 parameters. So that's why people started to estimate not M, to do averaging. Not M, but say determinant of M. If you work with the optimality. Or M minus 1 should be average. Then this phenomenon disappears. So it should be some kind of cautiousness in how you introduce averaging. Even if you are Bayesian or whatever, so you still have to be cautious. Because somebody in audience can notice.
Okay.
And this fact is actually, okay, so people notice it. And now, can we go to the next?
Can we answer to that comment?
Yeah.
Can he answer, or if he?
Did
I think
he work with you?
no. But so Sebastian, no, because I think what he's doing is indeed ED optimality. So I think he's averaging the determinants at the end.
No, actually,
It's for
But
everyone.
this slide was not about the averaging like a Bayesian or pseudo-Bayesian. This slide was more how to handle covariates when you have covariates and you, at the design stage you have to have a priori function for the covariate distribution. So
Yeah.
this is different than averaging across the parameters values.
So it's
here
not,
are covariates, are not
it's-
parameters.
do average with respect to covariates.
Yes,
But
covariates.
you see, it should be still the same phenomenon.
It's the same phenomenon for covariates. Yes.
So it's better to do, thank you for just correcting me, but it will be the same.
Okay.
but how would
So
you,
it's
how would
better
you,
to average determinant or inverse efficient.
Because your covariate could be 1 point, but you see, you do averaging.
Yes,
and
yes.
It's the same phenomenon.
okay.
So he took that from our paper, so that's why.
It's the same phenomenon. You have to be careful what to average. And now, if you move a couple here, you see what you are doing. So you're doing 2 steps procedure. You have empirical distribution, some histogram, whatever.
Yeah.
two-dimensional one.
Yeah. Yeah.
Then you approximate this empirical distribution with some nice smooth function.
Then you just
And now you do numerical integration. Whatever method you use is it, if, and in your case, it's Simpson quadrature. So it's you again going back to discrete problem actually, and do replace integration by summation. So you have two steps, and on each step you introduce some distortion with respect to reality. Why not just to do summation from the very beginning with respect to empirical function, empirical distribution function? It's what you know. The rest is fantasy or mathematical artifact.
Okay, that's a good point.
Okay,
Thank you.
There's another question.
I had a question about the dropout. You said that dropout will depend of MMSE or MMS status at baseline or changing with time?
No, at baseline. It was only assessed at baseline. Or no, not baseline actually. At the screening phase.
Okay. And so that's why it's also that it's independent of
Yeah.
the parameters.
Obviously,
It has different parameters, and you assume they are known.
yeah. So
You don't assume that you have to estimate
no,
them.
exactly. So that's also something we will improve. So obviously you gain information if you would try to estimate them simultaneously. But then that's nice, or then it becomes more complicated.
Well, Sebastian, it's really about the covariance again. I'm sorry. You got, from the paper, I got the impression you got two, the marginal distribution of age and the marginal distribution of MMSE. These will be highly correlated with each other. And
No, actually, I investigated it. It was not highly correlated. The correlation is 0.1 or something.
So from the paper, that they actually gave you the correlation structure between those two parameters.
No, we got it from the clinical. No, I don't have to go there, but from the clinical trial databases. So we have the raw data information.
So as people get older, their dementia doesn't get worse.
Say again, please.
Because you arguably speaking, you'd say that as someone gets older, their dementia gets worse. MMSE should drop down if there should be a negative, strong correlation between those two variables.
Yeah.
So I'm just surprised.
Yeah, I was surprised as well. But if we use the empirical distribution function, that will be solved automatically.
Because I just don't think that the marginals can be combined into a joint distribution without clear knowledge of that. Also, this will be a snapshot rather than a temporal thing following longitudinally somebody over time as well. Whereas your study is a 24-month study following somebody over time.
But it only informs about the baseline, the MMSE.
Okay.
Just like a generic question. You started out by telling us that the problem is that many of these phase 3 studies fail in Alzheimer. Your design doesn't really address that issue, right? We could perhaps try to address that issue by taking a perspective that, I don't know, there's probably like a whole sequence of such phase 3 studies for different drugs as they come through the pipeline. And that would then suggest an entirely different approach potentially, right?
Yes. One could do a more integrated approach that uses the information throughout the different phases. That's true. But we think that this is like a tool for the team that plans the study to do an informed decision about the next clinical trial. Because now it is really very much about traditions or convenience.
The same question in different words. You had that one slide where you showed like different stakeholders in the trial. And so if you take the notion serious that you want to do something about the failing studies, then you probably want to minimize the size of the trial so that you can start the next one, right? Which probably is like the sponsor's interest, right?
Yeah, I think it's not that easy.
What is it? What makes all these studies fail? Is there any systematic?
Yeah, I mean it is very small change over time. So you have a big placebo effect. These examinations, the people learn from that is also another thing. So I think it's at several points. That should be addressed.
Maybe earlier phases.
Yes, I think the drug doesn't work.
And maybe they don't have any good ideas.
No, but I think the drug doesn't work.
But then they probably shouldn't initiate the phase 3 study if they had honest judgment, right?
Yeah, so that's why if you optimize a phase 2 study, you get more precise parameter estimates, then it would help you to make a drug fail if there's no effect. So.
Oh, and if you were to take the right approach, then the optimal sample size should be 0. Don't even start it, right? Well, some.
It depends how much confidence you have in your drug, I think.
But really to comment, so nobody really does a serious problem when you try to somehow optimize the size of phase 2, for instance, where they do dose finding, and phase 3. So everyone puts a 50 or 100 patients on phase 2, and then 10,000 on phase 3, which is stupid, but nevertheless, it's traditional in pharmaceutical industry. This 100, especially with that type of variability, you saw.
And also, I've heard you talk about these trials by a physician in academia in US, and you think that the market is so big for drug companies that they don't care doing these big trials, even if they will fail. You know, if there is a little hint in phase 2 that it might work, they do it because they will, you know, stack first. It's not good for patients.
Not anymore.
Okay, so let's thank Sebastian again.